Good morning, students. Myself, Major Komal Katyal, Department of Zoology, GK Gowani Government College, Beenmal. Continuing my lecture on immunology or immune system. Today, we will discuss about antigen. आज हम बात करेंगे प्रतिजन के बारे में. Now, what are antigen and immunogens? The substances that can elicit an immune response, which is a humoral or a cell-mediated immune response when introduced into an animal. Although the two terms are used interchangeably, there are differences between the two. Any substance that binds to an antibody or a T cell receptor is known as an antigen, but some antigen cannot elicit an immune response. So, the immune response generator are used for the term immunogens. Now, what are antigens? The ability of an antigen to produce an immune response and to react with its product is known as the antigenicity. प्रतिजन की वह क्षमता जिसके द्वारा वह प्रतिरक्षी तंत्र में एक उद्दीपन उत्प्रेरित करता है और जो प्रतिरक्षी बनते हैं या जो उत्पाद बनते हैं उनसे वो क्रिया या अभिक्रिया करता है. The ability of a material to induce an immune response is known as immunogenicity, and such materials are known as the immunogens, which we have already discussed in my previous slide. The material which induces an immune response as well as reacts with the antibody is also known as a complete antigen. So complete antigen can be called as immunogens. While the material which reacts only with the antibodies but cannot induce an immune response is also known as a incomplete प्रतिजन. वह प्रतिजन जो केवल प्रतिरक्षी से ही अभिक्रिया करता है उसे अपूर्ण प्रतिजन कहते हैं. Now, what are haptens? Haptens are small molecules which are non-immunogenic. It means they are the incomplete antigens. Thus, could never induce an immune response by themselves. They may be able to bind to antibodies or T cell receptors. Now, it is derived from the word haptene, which means to bind. It is a Greek word. They become immunogenic on combining with large molecules, namely the carrier. It can be dinitrophenol. A hapten, when combines with serum albumin, it becomes a complete antigen. Some substances may act as hapten in some animals, whereas they act as complete antigens in others. For example, pneumococcal polysaccharide may be a complete antigen in mouse and man, but it acts only as a hapten in rabbits. इस तरह से हम समझ सकते हैं कि हैप्टेन बहुत छोटे अणुभार वाले पदार्थ होते हैं जो इम्यून रेस्पॉन्स उत्पन्न नहीं कर पाते, पर उनको किसी न किसी कैरियर या माध्यम की आवश्यकता होती है. Now, the term epitopes and paratops. Epitope, the portion of an antigen that is recognized and bound by an antibody or a T cell receptor, that is TCR, is known as an epitope. Epitope means the antigenic determinant. Paratope, the portion of the antibody molecule that binds to the epitope, is known as a paratope. Now, there are two kinds of epitope. One, the B cell epitope, which is recognized by the B cells or the B lymphocytes, and the T cell epitopes, which are recognized by the T cells. Usually, epitopes are found on the surface of the antigen, but unfolding of the antigen has revealed that the antigen has hidden epitopes apart from the functional surface epitopes. So they have two types of epitopes. One, the functional surface epitopes, and the other ones are the hidden epitopes. Now, the one protein may have multiple antigenic determinant. It means the number of epitopes may be one too many in an antigen. The number of antigenic determinants per molecule is referred as its valency. For example, BSA, the bovine serum albumin, has total valency of 18. It means it has total 18 epitopes, which includes the functional, that is the surface epitope, as well as the hidden epitopes. It means it has 3 times more valency than its functional valency. Now, the immunogenicity, it is the ability to induce a humoral, that is the antibody, or a cell-mediated immune response. We can classify the immunogens into two categories. One, the weak immunogens, and the second one, the strong immunogens. The weak immunogen example is given as insulin, and the strong immunogen example is given as hemocyanin. Now, what are the essential factors for immunogenicity or antigenicity? First, the foreignness. It is essential for immunogenicity, which is self-responsive immune cells are eliminated during lymphocyte development. The more distant or foreign the antigen source, the better the antigenicity will be. For example, plant proteins are good antigen for animals than the duck serum proteins, which are basically of animal origin. Second, the size. It means the molecular size or the molecular weight of the molecule. The molecular size of an antigen has direct relation to the antigenicity. For example, hemocyanin with a molecular weight which is 6 into 10 to the power 6, and thyroglobulins which has molecular weight of 6.69 multiplied by 10 to the power 5 are excellent antigens because of greater molecular weight, whereas insulin with a molecular weight of 5700 and histone with a molecular weight of 6000 are poor antigens. The chemical composition, as far as the chemical compositions is concerned, proteins are always better antigen than nucleic acid, while nucleic acids are better antigen than polysaccharides, and polysaccharides are better antigen than lipids. As far as structure is concerned, the primary, secondary and tertiary structure also play some role in determining the immunogenicity or antigenicity of the substance. The last of the essential factor is their physical form. The particulate has more antigenicity than a soluble form. Now, what are the host factors which affects the immunogenicity? The difference across a species, that is the interspecies differences. Difference within a species, it means the intra-species differences. The responders and non-responder to vaccine. Second, the differences in disease severity in epidemics. We can understand it by the following examples, that some human responds properly to the vaccine while others not, and at the time of epidemics some severely affected by the epidemic while some not. It depends upon how weak or how strong their immune system is. their genetic constitution, आनुवंशिक लक्षण भी इसको प्रभावित करते हैं. Their age, generally the greater the age, it means older the people, their immune system becomes weak. Now, what are the types of antigens? Isoantigens are the antigen present in some but not all members of a species have that antigen. For example, our blood group system, which is the ABO system, it consists of two antigen, one antigen A and other antigen B. And in some human, only antigen A is present, while in other only antigen B is present. In some, both the antigen are present, and in some, the antigens are totally absent, and on the basis of which we classify the blood group into four categories: the blood group A with antigen A, blood group V with antigen B, blood group AB with both the antigen, and blood group O, which is 0. It means there is no antigen. So these blood group antigens form the basis of blood grouping are cross-linked antigen. The major histocompatibility complex are the cell surface glycoproteins. Now, the heterophile antigen or the heterogenetic antigens, the antigens which exist on the surface of tissue cells of unrelated plants, animals and bacteria, but are identical to that antibodies to one will cross-react with other. For example, the human blood group A antigen is cross-reactive with antibodies of pneumococcal capsular polysaccharide type 14. Other example is the human blood group B antigen is cross-reactive with antibodies to some strain of E. coli. Now, continuing with our topic on types of antigen, they can be broadly divided into two: the endogenous and exogenous. Endogenous are those antigen which originate in our body, and exogenous are those antigen which are foreign in origin but circulate within the body. They may be a bacteria, a virus or a fungi. कहने का मतलब है जीवाणु, विषाणु और फफूंद. The endogenous are further divided into two categories: the autoantigens and alloantigens. Autoantigens are generally present in all individuals of the same species. Say for example, nucleoproteins. As we know, nucleoproteins are present in all species, all members of a species. The alloantigens actually reflect the genetic polymorphism, as we have already discussed about the ABO antigens, and the HLA antigens is the other example of alloantigen. Now first about the autoantigens. The autoantigens are substances capable of immunizing the host from which they are obtained. Self-antigen are ordinary non-antigenic. It means those antigens which are present in a particular individual cannot initiate the immune response. Modification of self-antigens are capable of eliciting an immune response. It means sometimes some modification takes place in your body's physiology and it induces the immune response. For example, the body lupus, the systemic lupus erythematosus. Now the term cryptantigen, which are the hidden antigen. An antigen on the surface of a red cell or red corpuscle that is not normally visible. It means it is hidden or in a crypt. These antigens are exposed either through the action of enzymes secreted by bacteria in acquired polyagglutination or as a result of an inherited defect leading to altered structure of the normal red cell antigens. Normally, the benign antibodies against cryptantigens are present in most human serum. An exposure of the usually invisible cryptantigen may result in polyagglutination. Examples are given in the slide T, TN and CAD. So this finishes our lecture on antigen. Thank you.